Hello, welcome to this web class. Just give me a quick thumbs up or emoji if you can hear me okay. Just check everything's up and running. Hi, perfect. Cool. Well, thanks for joining, guys, particularly seeing as it's a nice summer's evening. It's good that you've chosen to study some biology. Where is everybody dialing in from today? Where are you guys based? I recognize some of these names from some of the classes. Some of you guys have been before. Hull, nice. Zara, yeah, using your sister's account. I remember that. Got some Londoners, nice. I'm currently in London as well. So what else? Okay, so all from the UK so far, but all over. Newcastle, Leeds, nice. A couple of Wolverhamptoners. Oh, we've got someone from Turkey. Nice. Cool. I think let's make a start. Yeah, as you guys are aware, this is a live session, so please feel free to interact. You can. I'm going to ask questions throughout the class, and yeah, the more you interact with it, the more you'll get out of the session. So yeah, feel free to leave comments. So let me just share my screen. So hopefully you guys can all now see a PowerPoint. Oh, I just need to make sure I can still see your comments. Okay, nice. Mohammed, currently on holiday in Bali. You jammy. That's yeah. Lucky you. Very jealous. So yeah, we're going to look at genetic diversity today. It's an area of the spec that it's not overly difficult, but there's just a lot of definitions that you need to learn. And yeah, students sometimes get some of the concepts mixed up. So we're going to just go over them. And yeah, you'll see it's just a lot of defining the terms, and that's how you do well in this topic. So a little bit about me. My name's Alex. I'm the head of biology here at SnapRevise, and I've been tutoring and teaching biology for the last 4 or maybe even be like 5 years now. A little bit about SnapRevise. So the reason we're doing this class on YouTube today is to show you guys a little bit about a little taste of our tutoring package. So we have tutoring on our website, and we do these classes like 5 sessions a week. So if you enjoy your session today, we're giving away a 1-week free trial for all of you guys, so you can come check out the website and yeah, give it a go. It's yeah, it's helped a lot of students with their studies. So those of you who've done one of these classes before, you might recognize this. We normally start the biology sessions with a little learning pyramid. So it's some of the topics, some of the questions from the topics we've been looking at recently in the classes. So you'll see they're not necessarily all related to the topic we're doing today. It's just a chance to keep stuff fresh in your guys' minds. So first question, what are the sources of variation in meiosis? You guys got mocks tomorrow. Nice. And then are you going to be mocks tomorrow, and then are you done basically for the rest of the term? And yeah, lots of guys got the correct answer: crossing over and independent segregation. Independent segregation. Okay, next one. What is meant by independent segregation? So you guys have said it here, but what do we actually mean by that? I'm extremely hot. This is all exam boards. So it is AQA and it's helpful for all of them. Sometimes you might be familiar with this as independent segregation, or you might refer to it as independent assortment. Some of the exam boards use one and some use the other one, but they're both synonyms. So you can use either one really, but best use the one in your exam board. Yeah, so basically what you guys are saying is good. So it's the random division of the chromosomes. So you've got maternal and paternal chromosomes, and during meiosis and independent segregation, they line up and they're separated. And yeah, crucially, it's completely random. So you could end up with the maternal or the paternal chromosome. This is AS, yeah, meiosis. Random, oh, the weird little glitch there on the screen. Random sorting of maternal and paternal. Nice. So humans have got 23 chromosomes. So there's 23 times you can either get the maternal or the paternal chromosome. So this creates variation. So the number of different options in humans is 2 to the power of 23, which is a big number. Other organisms have more chromosomes. There's even more variation. Some have less, and it'd be slightly less. So I think this corresponds to something like 8,350,000. I think it's something around that. So yeah, a lot of different possible combinations in humans. So next question, at what stage of meiosis are haploid cells first produced? Seems like loads of you guys have got mocks next week. Are you all from the same school, or just a coincidence? Cytokinesis 2, close. It is cytokinesis, but it's of cytokinesis 1. So the end of, end of prophase 1. Of prophase 1. Cool. Next question, what is the purpose of cytokines? I think some specs don't really go into this. So if you haven't heard of cytokines, then don't panic. It might not be in your specification. What's haploid again? Haploid is a single set of chromosomes. So normally we have chromosomes in pairs. We have 23 pairs. A haploid cell just has 23, not 23 pairs. It's essential for sexual reproduction to make gametes, which are haploid, because you need 2 cells to combine. And if they weren't haploid, every time you had fertilization, you'd be doubling the number of chromosomes, which would be getting pretty ridiculous after a few generations. So yeah, you make haploid cells for gametes. Yes, nice. So they're used as cell signaling molecules. They can activate other cells. So some of you guys have said they trigger the production of antibodies or T cells. So yeah, they can be used to gear up the immune system. Yeah, so what should we put? Cell signaling plus activating cells. Okay, now throwback to enzymes. So how do competitive inhibitors different differ, sorry, to non-competitive. Yeah, nice. So competitive bind to the active site, and non-competitive bind to a different part of the enzyme known as the allosteric site, as some of you guys have mentioned. And that changes the shape of the active site. So the reason we call them competitive is because they compete with the substrate. So yeah, they have to have a similar shape to the substrate, whereas a non-competitive inhibitor could be a totally different shape to the substrate. Yeah, nice. So I can't see, I don't think anyone's mentioned about permanentness. So generally, competitive are, it's temporarily blocking the active site. Often allosteric ones can be permanent, but there is temporary and permanent ones of both. Different binding sites. And what else are we going to say? Only competitive, similar to substrate. So inhibiting enzymes is really important because sometimes we want enzymes to be working, and then other times we want to basically be able to turn them off. If you don't use inhibitors to turn off an enzyme, you would have to completely destroy it and then make it from scratch each time. So that would be quite inefficient. So we can use inhibitors to prevent that process. So final question, what can you guys remember about selection? We're talking about, presume we're talking about natural selection here. But we're also going to look at some other types. Yeah, nice. Nikki has got all of the types there, natural selection, and then the 3 different types of selection, which we'll go through at the end. Cool, nice. Yeah, should be with the same answers. Sexual selection is another form of selection. Yes, very true. This is mostly AS content for this lesson. In fact, no, the whole thing is AS. Yeah. Okay, so these are the objectives from this class. We're going to look at what is meant by genetic diversity. We're going to describe how evolution occurs, and we're going to look at the different types of selection. And that's what you guys were just talking about. What we won't go into today, but what is in the spec is the evidence for evolution. So this is things like fossil record, etc. But yeah, we're not gonna cover that. We can't do everything in an hour. So these are the spec points. I'm just going to flick through them quickly because you guys have presumably got a copy of the spec. If you haven't, you should download one. They're really useful. You can just get one if you just go onto Google and type in your exam board and the specification. You can get that, and you should be referring to them quite regularly because it's really important to know what exactly you need to know. The biology syllabus is so big, and you've got to really, yeah. I don't want to discourage extra reading around the subject, but at least make sure you know all the spec, and then you can do some extra reading as well. So first up, genetic diversity. Can you guys give me a definition for genetic diversity? It's probably one, you've probably heard the term, but maybe a little bit tricky to actually define it. So let's see what you guys can come up with. This session, yeah, it's an hour long session. So we'll finish around 6 o'clock. Can you relearn all of the year 12 content between now and September? Yeah, definitely. That's plenty of time to go over all the topics. Yes, so it's the total number of alleles in the population. So you need to use the term allele here, not gene. In a population. So it's really common to write gene in the definition here, but you actually would lose the mark for that. So you need to be specifying alleles, which are a version of a gene. Yeah, so it's specifically that. But it looks like most of you guys said allele, so that's nice. So the more alleles you have, the more genetically diverse that population is. And this is important because genetic diversity increases the survival rate of a population. So if you've got low genetic diversity, it means you haven't got many different traits or different alleles in that population. So if the conditions were to change, for example, due to climate change or maybe a new disease comes about or a new predator, that those organisms with a low genetic diversity are going to more likely to go extinct. There's a lower chance of having the suitable allele to cope with the changing conditions. So yeah, more genetic diversity is better. A lot of conservation programs, we need to be like really aware of making sure we maintain a good level of genetic diversity. Otherwise, the program ultimately won't be very successful. So what we're putting here, more GD equals higher chance of survival. Not sure why I put a space there. Population survival. Greater probability of an organism having a suitable allele. Cool. Does this link to genetic bottlenecks? Yeah, we're going to be talking about those later. Yeah, and the founder effect, exactly, which is pretty similar. So, in fact, literally right here. So right on cue. This is two more terms you need to know. So can you guys define a genetic bottleneck and the founder's effect? They are similar, but slightly different. The effect is the same, but they sort of occur in different places. So what are these things? Do you need this for OCR? Yeah, I guess we're pretty close to the spec still, so we can just double check. Yeah, so genetic bottleneck and founder effect is in OCR. I think it's in all of the specifications. So yeah, possibly not in Edexcel A actually. Doesn't seem like they have that much on this topic. Yes, nice. So a genetic bottleneck is an event that reduces the gene pool. So it's usually some sort of catastrophe that wipes out most of the population. So you've only got a few individuals left. Therefore, there can't be that many possible alleles in the population. So if you repopulate from that small group, you can get a different makeup of the gene pool, and it's going to have a lower diversity. So we quite often model this with different colored marbles or beads representing the different alleles. So this is the population pre-catastrophe. Maybe it's a flood or a fire, something like that. Then there's a catastrophe. Only 2 of the whole population survive. So that's the alleles that are remaining. and then the repopulate from that, we've now got a low genetic diversity. So the population might reach the same size as it was before. Usually there's a certain carrying capacity for that environment. So unless it's been drastically altered, it will probably regrow to the same size. But that population has a lower diversity. They're going to be less able to adapt to changes. So yeah, greater extinction risk. The founder's effect is essentially the same thing, but it's specifically for a new area. So this is a common thing to occur on islands. So if you start a population from a few individuals and grow it from there, again, it's going to have a low diversity. So like, well, I was about to use Darwin's finches in the Galapagos as an example, but actually that's probably not a great one because we also talk about that with natural selection. Yeah. Is this in AQA? Yes. Yeah. I think it's in all of them. So what are we gonna say here? Sharp reduction in genetic diversity following a catastrophe. And then the Fans effect. So when a small establishes I kind of want to use the word population again. What's another word? I guess a community in a new location. So both of these reduce genetic diversity. Reduce GD equals higher extinction risk. So there are some examples of this in humans. So the Amish people in America, they were founded from a population, I think it was a starting population of around 200 individuals, maybe slightly less. And they quite strictly tend to marry within their population. So essentially there's been many generations of inbreeding within a small gene pool. So there's lots of problems with recessive diseases, recessive conditions. That's another risk of having a low genetic diversity. It makes the population more susceptible to recessive diseases. It's recessive diseases that are the problem because dominant diseases would be showing anyway, but recessive diseases you only get when you have 2 copies of the allele. So they're the ones that are like the risk of those goes up when you inbreed, which is why pedigree dogs are typically a lot less healthy than like non-pedigree dogs, which have a greater diversity. Cool. Oh yeah, some of you guys mentioned the Amish. Yeah, nice. What's another example? There was some people in, I think it was Kentucky, also in America, who they had a recessive disease. Oh, I don't know if we call it a disease, but it made their skin blue. So it was I can't remember what they were called now. I think they gave them some sort of name. But yeah, that's another one. You could talk about, yeah, anything on an island. So when something gets to a new island and colonizes it from there, that's the founder's effect. So it could be a tortoise drifts out on some like driftwood. They can go for quite a long time without water. So they've managed to reach lots of islands, even though they don't actually swim. They just get like blown offshore. Birds are quite good at colonizing new islands. Plants can get there from either like a coconut floating in the ocean, or the seeds can go in the air. So yeah, that's all examples of founder's effects. The more remote an island is, the lower the genetic diversity, because there's probably going to be less founding individuals. Okay. A genetic bottleneck. What can basically, an example would be like a disaster, a natural disaster that wipes out 95% of the population or more, something around that, and then you have to rebuild from that. The Fugates of Kentucky. Yes, thank you. That's the one. You don't really need to know examples, but you could just give, you could basically make one up. Talk about any animal on an island. That's the founder's effect. Talk about any natural disaster, reducing the population. And that's an example of a genetic bottleneck. So you could give like the, do you remember the big forest fires they had in Australia last year? That has reduced the genetic diversity of koalas. And they're actually worried that koalas now have such a low genetic diversity that they may go extinct. So that could be an example you could give. Yeah, there are both examples of how genetic diversity is reduced. So be very careful that you're talking about genetic diversity and not biodiversity. Biodiversity refers to the number of different species in an area. It's a good indicator of like a healthy ecosystem, or we tend to prioritize conserving ecosystems with a high biodiversity rather than like a low biodiversity. And it's one of the measures we look to see how well we're looking after an ecosystem. So if an ecosystem has whatever the relative biodiversity is, we're seeking to maintain that biodiversity and that's how we judge whether conservation is working correctly. What are the spotted moths? Yeah, the peppered moth. That's an example of natural selection. So that's it's a really a common one that comes up at GCSE as an example. So you guys have probably heard it from that. Yeah, it just showed how there was a change in the environmental conditions. So the Industrial Revolution, it made the trees go black. They were burning so much coal. All this soot it made the trees go black. Originally, they were birch trees, so they were white. So the peppered moths, initially, the black ones were selected against. They were a disadvantageous allele. So there weren't many fully black moths. Most of them were this sort of peppered white color, which blended in on the trees. But then the trees were turned black by the smoke. So the black moths were now selected in favor. They had an advantage. They were now camouflaged, and the white ones were the ones that were getting predated more by birds. So the phenotype of the population became, over a few generations, became like more black moths than white moths. So yeah, natural selection in action. Okay, so just a quick check. How are you guys feeling with that so far? Hopefully all good. And we'll move on to evolution. Nice. Cool. There's a bit of a lag between when I say something and when the comments pop up. It's like 5 or 6 seconds later. So I have to I ask something and then I don't see it for a little while. It's a bit weird. Cool, nice. Lots of 1s and 2s and the nice dabbing cat thing. Cool. Evolution. Evolution is, well, this was the theory first put forward by Charles Darwin, that things evolve due to natural selection. So you guys have obviously all heard of natural selection. Well, I presume you will have, if you're studying biology. You need to be able to sort of describe how the process works. And it's something that, again, students typically don't do this very well. There's kind of like a set way of sort of describing it. So I'm just gonna go through how you could structure a 4 or 5 mark answer on this. So yeah, that sort of question comes up relatively often, just like describe the mechanism behind natural selection. So how would we start it off? Has anybody got any ideas? or yeah, how do we begin? We are not going to do taxonomy and phylogeny in this session. No. Again, too much to do for just 1 hour. But so if you're enjoying the class today, we do way more sessions on our website. So yeah, we do like, I think we've got 5 classes a week at the moment. So we go through a lot more topics than we do on YouTube. Nice. Yes. So random mutation. Leads to new alleles. Nice. So this leads to anatomical, physiological, spelled with a Y, not an I, and behavioral differences. So anatomical is like different structures. Physiological is like the sort of biochemical processes in the cells, and behavioral is obviously well, how else would you define it? Like I can't define it without using the word behavior. A change in behavior, basically. Cool. So yeah, if the environment changes, the new alleles may prove to be an advantage. So one of the key things, one of Darwin's principles for natural selection is that there needs to be more offspring produced than can survive. That's like the survival of the fittest idea. So the ones with the best alleles for the environment are more likely to survive and then survive and reproduce. So over time, the population will change. So more offspring produced than can survive. Best, or yeah, let's go with that. Best adapted, most likely to survive and reproduce. So they will pass on their alleles and population will change over time. Will change, oops, let's just start it again. Over time. So yeah, something like that would be a sort of way to structure your answer. You don't have to do it like word for word like this. And this little bit is mostly AQA make a big deal out of anatomical, physiological, and behavioral differences. If you're not AQA, you could probably just say leads to differences. So you wouldn't have to worry about those 3 words. What does the end bit say? They will pass on their alleles and allele frequency in the population will change over time. Yeah, sorry, I have pretty messy handwriting, so it can be a little bit hard to read sometimes. If you ever need something translating, let me know. So just as a recap, well, we've got a picture of it here. What do we call this diagram that we've got? Yes, the allele frequency changes over time. So that's like, yeah, an important thing to put into into your answer for natural selection. Yeah, I don't know if it's, oh, there we go. Yeah, it's a phylogenetic tree, exactly. Nice. This shows evolutionary history. And we use them basically when there's a branch, when they split, that's when they last shared a common ancestor. So the more recent the split, the more recently they shared a common ancestor, the more closely they are related. So this is an example of some of the major animal groups. So we've got bony fish, cartilaginous fish, bony fish, amphibians, mammals, reptiles, and birds, and it's showing how they are related. So birds and crocodiles and mammals diverged from amphibians around here. And yeah, you can keep going back. So the first thing to evolve was amphibians. They're the first like land dwelling animals. So they split longer ago. So as humans, we'd be related to these. These are both mammals. And yeah, we're least closely related to cartilaginous fish, which are like stingrays and sharks. And yeah, more closely related to birds and reptiles than amphibians, etc., etc. What is that long word? Sounds like a medical condition, or is it just a made-up one? How did amphibians form birds? Yeah, from millions of years of natural selection. So amphibians were the first to evolve from fully aquatic organisms, and amphibians never actually fully shook off the need to have water. So amphibians lay their eggs in water. They have to lay their eggs in water, and they start their life cycle as tadpoles. So they live fully in the water initially. In the adult form, they can come out of the water, and some, for example, some toads don't really spend that much time in water, but they need to always be in a relatively moist environments because they breathe through their skin, so their skin is damp, so they lose a lot of water. And yeah, they need to come back to water to reproduce, so they can't live in certain environments. The next ones to evolve were the reptiles. They basically solve the problem of needing to have water by developing scaly skin, which they then don't lose so much water through their skin, so they can live in drier environments. And they developed eggs with hard shells that don't need to hatch in water, and the young don't need to be aquatic. So that enabled them to colonize new areas that hadn't yet been colonized. Then birds evolved from reptiles. Again, they have the shell on their egg, which means they don't have to be laid in water. And obviously birds developed flight. So they were able to colonize lots of areas. Reptiles had already developed flight at that point as well. They were flying dinosaurs, which went extinct with all the other ones. Oh, it's a lung disease. I see. Nice. That's a good one for Hangman, if you can remember it. Cool. So I kind of forgot what we were talking about. Oh, just natural selection. Yeah. Oh, and then mammals evolved from the early birds. So mammals don't lay eggs. They gestate their young inside their bodies, and they give birth to live young. Actually, some sharks give birth to live young, which is, even though they're like very, very distantly related to mammals, they share that in common. That's an example of convergent evolution, where two similar traits evolve totally unrelated to one, like to each other. So another example of that is flying. So there's lots of different flying organisms like dragonflies, or lots of insects can fly. Bats are mammals that can fly. Birds obviously can fly. They all develop flight individually. It doesn't necessarily mean that they're related. Try to pronounce that long word. Okay, pneumony ultramicroscopic volcanosis. That was my best effort. Like the crabs. Not quite sure what you mean there. Anyway, yeah, let's keep going through, otherwise we might not finish. So genetic drift. This is another thing that confuses people. Well, sometimes you model it with colored beads again. Don't know if this is going to be that helpful, but essentially all genetic drift is a change in alleles over time. Crucially, the difference between this and natural selection is it's not because they're favored. It's just randomly certain alleles might be selected for. So yeah, it's not because they're favorable. This only occurs in very small populations. So yeah, let's put some things down. So some alleles are chosen completely randomly. These will get passed on. To next generation. Reduces genetic diversity. And only in small populations. So this, crucially, this links back to founder's effect and genetic bottleneck, because both of these create small populations. So yeah, that means that genetic drift is more likely. So the two things are kind of interrelated. So yeah, it's not really stabilizing selection or directional selection. I guess, well, it's more like directional than stabilizing, I guess, if you had to pick one of the two. Yeah. So yeah, if you've got a small population, just due to like random chance, maybe you've got individuals with a certain allele, just due to random chance, bad luck, none of those individuals successfully managed to reproduce. In one generation, we've just lost that allele. If they all don't reproduce, and again, it can't be a disadvantageous allele. In that case, it would be natural selection. It has to just be randomly. They don't reproduce. That allele is gone from the population. So yeah, the genetic diversity has been reduced. This is kind of, I don't think it's that helpful, this diagram. This is showing each one is a generation, and what you can like physically model this. You might have done it in your classes at school. You literally just grab a handful of the beads, whatever colors you have. So maybe you grab 5 out of the jar. They're the ones that reproduce. The individuals that manage to successfully have children, their alleles are going to be what's in the next generation, and you repopulate from the 5 that you picked up. So just by chance, even though there's equal numbers of red and blue, you might pick up more blues than red. It's not that blue is better than red, it's just random chance. So then when you repopulate from this one, you're going to get more blues. This could happen a few times, and just due to random chance, one time you pick up a handful and there's only blue beads. and that's what you're going to repopulate from. The red bead, the red allele, is now completely gone from that population. So we've reduced genetic diversity, essentially. If the allele is completely gone, is that the reason some species are extinct? No, so this isn't the same thing as extinction. Extinction is where the species dies out. This is just reducing genetic diversity and changing the allele frequency in the population. So it doesn't have to necessarily reduce, like get rid of the allele, but it's at least going to have a different frequency than the original population. Speciation is when the alleles are too different. Kind of, yeah. You get disruptive speciation when there's like some very different ones. Is this the same as Charles Darwin? It's not the same as natural selection. Yeah, it's different. Natural selection is favorable alleles are selected for. This is randomly alleles might not get passed on. So yeah, the random element is in this, and it's only in small population, whereas natural selection is in all going on all the time. Interbreed means, yeah, breeding with other species, species breeding with other species. Inbreed means closely related individuals are reproducing with each other. So inbreeding increases the chance of recessive disease. Interbreeding creates hybrids in some cases. A lot of animals, you just can't, they won't have successful offspring, but some can interbreed to form hybrids. Any real life examples of hybrids? Yeah, there's lots. So a liger and a teon, that's when you've got a lion and a tiger that reproduce. The crucial thing for a hybrid is that the offspring are not fertile. So yeah, hybrids are always infertile. Otherwise, those two things are actually the same species. Because that's how we define what a species is. That's something that can reproduce and interbreed and produce fertile offspring. So yeah, if the offspring's fertile, they're actually the same species. A mule is another one. Yeah, exactly. So a mule is a cross between a donkey and a horse, and they are infertile. So they create mules because they have kind of some of the good qualities from both. They're particularly strong, but they get that from the donkey, and they require like less food and water than a horse, but they're much bigger than a donkey. They get that from the horse side. Yes, you get zonkies as well. They can cross zebras and donkeys, and they can do the same with horses. So I presume they just call that as a zorse, maybe. What else is there? There's a wolfen, which is a false killer whale crossed with a dolphin, and there are lots more examples. I can't think of any off the top of my head right now. The British royal family are hybrids. No, not hybrids, but they are the royal families of Europe are all very inbred. They've tried, or recently they've stopped marrying directly into their family, so the genetic diversity has been improved in the last few years. But before that, everyone was marrying each other's cousins basically for many hundreds of years. So they have real problems with recessive diseases in the royal family. So hemophilia was a big one for a while. A lot of the royals had hemophilia. Intra and interspecific means intra is between members of the same species. Inter is between members of different species. So you can have inter and intra-specific competition. Cool. How are we doing with that? Yeah, I guess you're right. So you can also, yeah, you can say it for members of the same species as well. Yeah, because that is how we use it in the definition. Yeah, no, good point. Nice. Cool. Let's do. I'm actually going to skip this question because it doesn't relate that much to what we've been looking at. And I kind of want to talk about the different types of selection before we finish. So yeah, if you want to do this, please feel free to screenshot and you can have a go after the lesson. But yeah, let's talk about selection. So yeah, there's three types of selection. Some exam boards don't talk about the third type. So I think AQA doesn't talk about disruptive selection. So yeah, just be aware. Anyway, yeah, so what are we looking at here? Who can tell me number 1 and number 2? What's the name of these two different types of selection? You should be able to recognize them from the shape of the graphs. So yeah, nice. This one is directional. And this one is stabilizing selection. So yeah, the example we've got here is fur length. So well, we should define what it is first. Basically, directional selection is where phenotypes at one extreme are favored. This means the ones with those extreme phenotypes are more likely to survive and reproduce. Therefore, over time, the population is going to move in the direction of that phenotype. So let's say there's some change in the climate. The climate gets a lot colder. For example, there's an ice age. So there's now colder climate. The wolves, let's pick wolves for an example. The wolves with the longest fur are now going to have an advantage. Before, it wasn't really an advantage. In fact, it might have been a disadvantage. They might have overheated. But now long fur means they can spend less energy on staying warm. Therefore, they're more likely to survive and reproduce. It's now advantageous. They're going to have more offspring with long fur. And again, their offspring with the longest fur are more likely to survive and reproduce, etc., etc. Over many generations, the population will evolve longer fur. They've adapted to the changing conditions. So yeah, that would be an example of directional selection. Another good example is antibiotic resistance. That's over time, the population is evolving to be resistant to antibiotics. So this happens very rapidly, and we're currently in the process of using antibiotics way too much, and a lot of bacteria are now resistant to pretty much all of the antibiotics that we have. So yeah, you might have heard of MRSA in hospitals is causing a problem because we rely so much on antibiotics for medicine that yeah, we kind of take it for granted that we have these, but we are within a few decades, unless we find new antibiotics, within a few decades of having diseases which we formerly eradicated come back because the antibiotics we use on them will no longer be working. Yes, high affinity hemoglobin. Exactly, that would be an example of a directional selection. If you move to a new environment, if you're in the same one, then that would be stabilizing selection. Yeah, nice. Yes, this is like types of like ways that natural selection takes place. Yeah. So stabilizing selection is where the phenotypes in the middle are favored. So one example is mass at birth, or you could also have like head size in humans at birth. So this is kind of like I don't know, like the Goldilocks sort of zone. Like you don't want to be too big or too small when you're born. That makes it less likely that you're going to survive. If you're too big, you might, well, potentially for humans, particularly if you're born and you're too big, there's a real chance that you could kill less now. Thankfully, we've got better medicine, but you could like kill your mother, and therefore, obviously, your survival chances are massively decreased if you haven't got a mother to look after you. So that's bad to have a big head. It's also bad to have a small head because you haven't got like room for much brains in there. So again, it's going to reduce your survival chances. So yeah, we kind of, there's a stabilizing selection pressure to have an averagely sized head when you're born. Hi Rush, how you doing? So yeah, what can we put for these ones? Individuals favored that vary in one direction from the mean. And stabilizing, individuals favored that are the mean, or yeah, let's put it like that. Yeah, I've heard of schools having problems with people finding the papers, and basically everybody gets 100%. So it makes it then very difficult to give people marks because you can't give everybody an A star. It like flags up to the exam board. So yeah, there's also disruptive selection, which looks like this. It's not in the AQA spec. So you've got this is the phenotypes, and over time, individuals, it's, yeah, like I said, it's not in all of the exam boards. Oh, has this frozen? I'm getting a, like it's spinning wheel of death. Oh, it looks like it's working again now. Cool. So, yeah, I think there's a few more. Let's quickly talk about these. So allopatric and sympatric speciation. This is where they're geographically isolated. And this is where they are not geographically isolated. So yeah, things, you can get speciation occurring when things are in different areas, like for example on different islands, or you can still get speciation occurring when things are in the same area. They have to just, yeah, there'll be certain things going on. But yeah, that's just two more. I said there's a lot of definitions and keywords for this topic. So yeah, that's just two more: allopatric, sympatric, disruptive, directional, stabilizing, genetic drift, genetic diversity. What else have we covered? Bottleneck, genetic bottleneck, founder's effect. Yeah, basically, if you make some flashcards with all of these, it's probably the best way to sort of revise them and learn them. Cool. So yeah, that is, I think we're going to stop the lesson there. So yeah, hopefully that was helpful, and you guys, yeah, learned some of those terms, and you can sort of go away and revise them a little bit more. Before we end, I just want to give you guys a quick demo of our website. So let's skip through this. Oh, and also we've got some more classes coming up. So set a reminder on YouTube. We do ones in other subjects. So we do physics, chemistry, biology, and maths. Obviously this was biology today, but if you do one of the other subjects, you can also join for those ones. So yeah, schedule a reminder and subscribe to our YouTube channel, then you won't miss out on any of the sessions. Yeah, so we do monthly ones on YouTube. We do way more sessions on our website. So we have a group tutoring package, essentially, where we currently have 5 classes a week. We go through AS and A2 topics. And yeah, you've got all the past recordings on there, but we do ones every week as well. So let's have a little look at the website. So this is our website. This is the package I was talking about. So we've also got like other stuff on our website. So have a look at it and see what it's all about. But what I'm here to talk to you today about is this tutoring package. So I'm going to go on the biology. They're done by Speciations. So this is the package. It's £50 a month. So this is for, well, at the moment you'd be getting 20 plus lessons a month. So way cheaper than tutoring. Tutoring is in London is like more than £50 an hour. Yeah, so this is a lot cheaper. It's obviously not one-on-one, but usually we have between, well, the A2 classes have been quite small at the moment because lots of the A2 people have finished for the year, so they're not coming to classes. But an average class would be anywhere between 5 and 10 students, sometimes a few more. So yeah, it's not one-on-one, but you're getting a lot of individual feedback. So you can ask questions, and unlike on YouTube where I can't answer everybody's questions, unfortunately, because there's too many students, the ones in our normal sessions, we can go through any questions and any things that you're maybe not sure on. You can like explain and make sure there's no misconceptions. So yeah, this is an example of a class I did at some point. So we have 2 different things. We've got web classes, which is like what we just did today. We kind of go through topics. We usually do more questions, but we didn't really do any today. Yeah, but we do introduce the topic, revise it, do some questions to make sure you know how to answer them. So that's web class. And we've also got drop-in sessions. These basically, we tend to go through lots of past paper questions. The crucial difference between these sessions is that you guys request these. So if there's a topic that you want to revise or that you feel you don't know very well, or you've got a test coming up in a particular topic, you just message in and we'll make a drop-in session for you on that topic. So we'll go through, we'll do a little bit of revision. Sometimes we do mind maps, that sort of thing, Venn diagrams, talk about it, and then answer some questions to make sure that you're happy with it. So yeah, that's those sessions. This is an example of, well, this is our schedule. So I've got some ones this week on, what date are we on? So we're starting with we're looking at aerobic respiration with A2, and we're looking at some photosynthesis stuff and exchange and transport, that sort of thing. Cool. What else to say? Yeah, we're doing, like I said, we're doing a 1-week free trial. So there's no sort of commitment required. So yeah, give it a go. Try out our classes for a week, absolutely free. If you then decide that you like it and you want to stick with it, then you can, at that point, you can commit. If you decide maybe, I don't know, you don't want to do it, that's also no problem. So yeah, here is the code if you want to do the trial. I think, well, it says it expires in 24 hours. So you need to do it like tonight or tomorrow if you want to sign up using this code. And yeah, I think that's more or less it. I'll hang around for another couple of minutes in case you guys have got any questions, and I'll do my best to answer them. Otherwise, yeah, thanks for joining and giving up your Sunday evening. And yeah, I hope to see you again in one of these YouTube sessions, or even better, in one of our like web classes on our website. Yeah, so have a good evening and hopefully see you soon. You can rewatch web classes. Yeah, yeah. So we record every session, and all the like the back catalog from like 2 years worth of lessons, they're all on there. So if you signed up, you get access to all the ones that have happened previously, not just the ones that you're there for. Yeah, no worries. You're very welcome. Yeah, nice. Any other questions? I just realized I made a tea, but I totally forgot to drink it. I'll have it now, but it's cold. Cool. Well, yeah, I think we've got no more questions. Trying to salvage your A levels. Yeah, I'll do my best to help with that. Right. Well, yeah. Have a good evening. Hopefully see you guys soon. And yeah, use the code tutorial7 to give it, to sign up and get a week's free trial. Nice guys. All right. Bye. Bye.